Hi, and welcome to this series exploring landmark experiments in protein science based on my book of the same name, and that link is in the description. As we saw in my video on the discovery of DNA, the structure of DNA was elucidated in the early 1950s, along with the proposal of an elegant mechanism for replication, and although most agreed that DNA was the carrier of heritable information, how it performed this function was still unknown. In contrast, the function of another type of nucleic acid, RNA, was largely unknown, although it had been noted that the amount of RNA a cell contained was correlated to the cell's degree of protein synthesis, and it was correctly suggested that RNA had something to do with protein synthesis. And so in this video, we'll have a look at how the mechanism of protein synthesis was elucidated. In the late 1930s and early 1940s, Claude broke cells open and separated the nucleus from the cytoplasm, which is what had previously been called protoplasm, and it's the contents of a cell outside the nucleus, by centrifugation, and noticed the material forming opalescent preparations which, in the dark field microscope, appeared to be composed of extremely small bodies, highly refringent and inactive Brownian movement, which is basically random movement. The size of the particles had been estimated to range approximately between 50 and 200 nanometers in diameter, and 1 nanometer is 1 million times smaller than a millimeter, with no apparent segregation in definite size groups. The isolated particles have been shown to be complex formations in which a nucleoprotein of the ribose type occurs in association with a definite proportion of lipids, especially phospholipids. The evidence so far indicates that the mass of the small particles does not derive from the grossly visible elements of the cell, but constitutes a hitherto unrecognized particulate component of protoplasm, more or less evenly distributed in the fundamental substance. In order to differentiate the small particles from the other already identified elements of the cell, it may be convenient in the future to refer to this new component under a descriptive name which would be specific. For this purpose, the term microsome, meaning small body, appears to be the most appropriate. Then, using electron microscopy, which, as the name implies, is a type of microscope that uses an electron beam instead of light to look at samples, he captured images of cells which revealed a lace-like network of slightly higher density than the rest of the cytoplasm, which was suggested to consist of vesicles and tubules interconnected in a continuous network. Since the network was restricted to the inner or endoplasmic region of the cytoplasm, Claude proposed the name endoplasmic reticulum for the entire structure, endoplasmic meaning inside the cell and reticulum network, and this is abbreviated the ER. The discovery of this network provided a likely candidate for the role of microsomal precursor within the intact cell, and it was speculated that the microsomes might represent derivatives of the ER, but the assumption could not be verified at the time because the electron microscopy of cells and tissues was still in infancy. In the 1950s, George Palade took advantage of improvements to the electron microscope and captured images which have demonstrated that in the dimensional range of the microsomes of cytoplasm possesses a network of membrane-bound vesicles and tubules described as the endoplasmic reticulum. Within this system, there are differentiated portions in which small, dense particles, 10 to 15 nanometers in diameter, appear to be attached to the membrane of the reticulum. Although this association is commonly encountered in certain cell types, in many other types the particles mentioned occur freely scattered in the cytoplasmic matrix. And Palade proposed that the microsomes identified by Claude were derived mainly from the endoplasmic reticulum, and that their characteristically high RNA content is due to associated small particles. Palade also noted that the two components concerned, i.e. the membrane of the ER and the particles, are fundamentally distinct structures. These dense particles rich in RNA were named ribosomes in 1958. Around the same time, Tor Hultin in Stockholm wanted to determine if protein synthesis proceeded in a specific order and in particular structures. So he injected the amino acid glycine labeled with N15, which is a radioactive nitrogen atom, and as I mentioned in previous videos, the presence of radio-labeled molecules in a sample can be detected with a Geiger counter. And so he injected N15 glycine into newborn chicks and periodically assessed how much N15 was incorporated in the nucleus, in mitochondria, in microsomes, and in the cytoplasm of liver cells. And his results are shown here, and we can see that N15 was incorporated into microsomes before any of the other fractions. And as expected, microsomes had the highest RNA content. Hultin noted that it is of special interest that the present experiments indicate that the ribonucleic acid is related not only to the transformation of polypeptide chains into proteins of specific structure, but also to the initial incorporation of amino acids into peptide chains. Since the microsomes are especially rich in ribonucleic acid and the production of phosphate-bound energy is centered in the mitochondria, the experiments indicate that the immediate availability of ribonucleic acid is more important for the formation of proteins than a close proximity to energy-producing enzyme systems. Around the same time, several groups found that protein synthesis could be inhibited by ribonuclease, an enzyme that specifically cleaves RNA, and it was stated that we can thus from now on consider the correlation between the ribonucleic acid content and protein synthesis as a well-established fact, and we can draw the conclusion that ribonucleic acid somehow takes a direct role in protein synthesis. In the early 1950s, Zemechnik and Keller were investigating the requirements for amino acid incorporation into nascent proteins and discovered a protein fraction from rat liver extracts that was required. Then, Hultin found that when ATP and pyrophosphate were incubated together in the presence of this fraction, the radioactive pyrophosphate was exchanged with the non-radioactive pyrophosphate in ATP, and this increased significantly when amino acids were added. In addition, he found that the increase in exchange was proportional to the number of amino acids added. And so this column is the number of amino acids added, and this is the percent exchange. And we can see that the increase in exchange was proportional to the number of amino acids added. Then, he used hydroxylamine as an acetylation trap to detect activated amino acids, that is, amino acids that were primed for insertion into proteins. And the acetylation trap is similar to what was used in my video on glycolysis and the Krebs cycle, but in this case, hydroxylamine was used as a trap instead of sulfanilamide, and so when amino acids are activated, they irreversibly bound to hydroxylamine, which turned a bright purple color when an iron salt was added, and these were the results of this experiment. And we can see that without adding hydroxylamine or amino acids, the hydroxymate formed was at 0, and this is how much ATP was lost and phosphate was formed. And in this case, ATP lost means that it's been hydrolyzed to ADP or AMP, and phosphate was released. If hydroxylamine was added by itself, then we get a little bit of an increase in hydroxymate and a decrease in ATP and phosphate formed. And this is an important control condition because it tells us that the hydroxylamine reacts to something by itself, and this needs to be subtracted from the results. If amino acids were added without hydroxylamine, then nothing really changed. But when both amino acids and hydroxylamine were added together, we can see an increase in the hydroxymate product and ATP lost, and an accumulation of orthophosphate equal to 2 times the amount of ATP. These results led Hultin to propose the following tentative formulation of the process by which amino acids are activated for protein synthesis. If E1 is an activation site on an enzyme for a particular amino acid, AA1, this site binds ATP in such a way as to labelize the AMP pyrophosphoryl linkage, which basically means to make it more reactive. The amino acid then displaces the pyrophosphate. These two equilibria would permit pyrophosphate ATP exchange to occur only in the presence of amino acids, would account for the failure of AMP to inhibit the reaction or to exchange with ATP, and the failure of pyrophosphate to accumulate in the absence of an acceptor. The additive effect on exchange of different amino acids suggests a separate activation site for each. In other words, these reactions don't result in any changes in ATP or pyrophosphate since they are both reversible, and the amino acid can be bumped out by the pyrophosphate and ATP regenerated. And this explained the lack of radioactive pyrophosphate accumulation in the first set of experiments, where adding more amino acids simply led to more p32 incorporation in ATP, since the pyrophosphate that was incorporated in ATP could be either normal pyrophosphate or radioactive pyrophosphate. But in the presence of hydroxylamine, the carboxyl-activated amino acid reacts to form the hydroxymate, which is irreversible, while AMP and pyrophosphate accumulate. And Hultin offered this figure to represent the reaction, where E represents the enzyme responsible for the reaction, of which there was 1 for each of the 20 amino acids. An amino acid and ATP bind to the enzyme, which catalyzes the bond between AMP and the amino acid with the release of pyrophosphate. And the fact that a similar mechanism was found in a variety of bacteria indicated that this process was likely to be universal. The next year, Hultin, Zemechnik, and their colleagues found that cytoplasmic RNA could bind amino acids and transfer them to microsomal proteins, and the binding of amino acids to RNA is shown here, where they synthesized the amino acid leucine with the radioactive carbon-14 and determined how much was incorporated into cytoplasmic RNA by assessing radioactivity, and we can see that adding more leucine to a mixture increased its association with RNA. Then they found that this process was dependent on ATP concentration, in a process similar to what they found for the activation of amino acids, and we see here that the more ATP was added, the higher the radioactivity was in RNA, and that adding a pH of 5 supernatant fraction of rat liver extracts to the mixture increased incorporation of leucine into RNA. The authors also found that adding RNase to the mixture, which is an enzyme that degrades RNA, didn't prevent the ATP pyrophosphate exchange or the amino acid hydroxymate formation, but obviously it prevented the incorporation in RNA, and this suggested that binding of amino acids to RNA came after their activation. To further elucidate the sequence of events in this process, they added RNA-bound amino acids to a mixture and assessed the amino acid content in RNA and microsomes, and here we see that over time, RNA loses its amino acid content, whereas microsomes increase theirs, indicating that incorporation of amino acids into microsomes happens after they're added to RNA, and that this was very efficient since it was essentially complete in 5 to 10 minutes. And finally, they found that this type of RNA, which is called transfer RNA or tRNA, only made up 2% of total cellular RNA, so they concluded that the evidence presented supports the conclusion that there occur ATP-dependent enzymatic reactions between ribonucleic acid and amino acids. These reactions are catalyzed by an enzyme preparation which is known to activate the carboxyl groups of amino acids in the presence of ATP. The product formed, an RNA or ribonucleoprotein to which amino acids are apparently covalently linked, is capable of interacting with enzymatic components of the activating enzyme preparation to affect the transfer of amino acid to peptide linkage in protein. It is therefore suggested that this particular RNA fraction functions as an intermediate carrier of amino acids in protein synthesis. Since the amino acid activation reaction is insensitive to ribonuclease, and since an activating enzyme has been isolated relatively free of RNA, it is still necessary to invoke an initial enzymatic activation reaction as originally postulated, followed by a transfer of amino acid to linkage on RNA. We have suggested elsewhere a hypothetical reaction sequence for protein synthesis which accounts for the findings presented in this paper. Its central idea is that pH 5 RNA molecules, each charged with amino acids in characteristic sequence, polymerize in microsomes in specific order determined by the complementary structure of microsomal RNA to higher molecular weight units with resultant configurational changes which permit peptide condensation between contiguous amino acids. Around the same time, Vulcan, Astrachan, and Countryman determined that there was an increase in RNA synthesis following infection of bacteria by bacteriophages, which are viruses that can infect bacteria. And these are the viruses in question, and we can see them infecting a bacterium in this image. And so the authors infected bacteria with viruses and then added p32 labeled phosphate for 6 minutes, and so any newly synthesized DNA and RNA will have p32, and then they added an excess of unlabeled phosphate so that from then on newly synthesized DNA and RNA will incorporate unlabeled phosphate, and in this way changes in DNA and RNA synthesis can be assessed. And so here the x-axis is time after infection, and they added p32 here and the excess of unlabeled phosphate here, and as we can see, RNA synthesis is greatly increased following infection, and here it goes back down because this is where unlabeled phosphate was added. And most surprisingly, they found that the nucleotide sequence of the newly synthesized RNA was similar to the virus's DNA rather than bacterial DNA, which suggested that bacterial DNA was quickly degraded and viral RNA was synthesized, noting that the similarity between RNA and DNA synthesized after infection suggests a hitherto unsuspected connection between RNA synthesis and DNA synthesis. The fact that RNA was quickly turned over also suggested that this type of RNA was unstable, in contrast to tRNA or ribosomal RNA, which were shown to be stable. And as we just saw, tRNA is the RNA that helps amino acids get incorporated into growing peptide chains, whereas ribosomal RNA is the RNA in the microsomal particles discovered by Palade. In 1960, Nomura, Hall, and Spiegelman performed experiments to determine where newly synthesized RNA was localized. Here, the authors added p32 to uninfected or infected bacteria for 7 minutes before extracting the ribosomal RNA. Then, they separated the samples by starch column electrophoresis, which is similar to what I talked about in my video on the discovery of DNA, where Chargaff separated nucleotides to determine the composition of DNA. And so here are the results. The x-axis is how far the samples ran in the gel. The left y-axis is the assessment of optical density using ultraviolet light, and this was used to assess all RNA since it was known that nucleotides absorb UV light, and the right y-axis was the assessment of radioactivity, which assessed newly synthesized RNA. And we can see that when bacteria are not infected, all RNAs are the same, whereas following infection, newly synthesized p32 labeled RNA had a different electrophoretic mobility than the non-labeled RNA, and this suggested that there must be a difference in nucleotide composition between the samples, and subsequent analysis of the sample's nucleotide sequence confirmed this. At this time, protein synthesis was thought to proceed using stable RNA molecules permanently attached to ribosomes, which required one ribosome RNA pair for each gene encoded by DNA. But evidence against this hypothesis was growing, and the following characteristics for RNA templates were suggested by Jacob and Monod in 1961. The candidate should be a polynucleotide. The fraction would presumably be very heterogeneous with respect to molecular weight. It should have a base composition reflecting the base composition of DNA. It should, at least temporarily or under certain conditions, be found associated with ribosomes, since there are good reasons to believe that ribosomes are the seat of protein synthesis. And finally, it should have a very high rate of turnover. They noted that the new species of RNA discovered by Vulcan and Astrachan met these specifications and named it messenger RNA, or mRNA, and the authors proposed the following mechanism for protein synthesis. The molecular structure of proteins is determined by specific elements, the structural genes. These act by forming a cytoplasmic transcript of themselves, the structural messenger, which in turn synthesizes the protein. The synthesis of the messenger by the structural gene is a sequential replicative process, which can be initiated only at certain points on the DNA strand. The same year, Brenner, Jacob, and Meselson proposed 3 hypotheses that could explain how RNA synthesis in E. coli switched from bacterial to viral following an infection. Model 1 is the classical model. After infection, the bacterial machinery is switched off, and new ribosomes are then synthesized by the phage genes. Model 2 assumes that in the particular case of phage, that proteins are assembled directly on the DNA. The new RNA is a special molecule which enters old ribosomes and destroys their capacity for protein synthesis. At the same time, synthesis of ribosomes is switched off. Model 3 implies that a special type of RNA molecule, or messenger RNA, exists which brings genetic information from genes to non-specialized ribosomes, and that the consequences of phage infection are twofold. A. To switch off the synthesis of new ribosomes, and B. To substitute phage messenger RNA for bacterial messenger RNA. This substitution can occur quickly only if messenger RNA is unstable. The RNA after infection does turn over and appears, therefore, as a good candidate for the messenger. It is possible to distinguish experimentally between these 3 models in the following way. Bacteria are grown in heavy isotopes so that all cell constituents are uniformly labeled heavy. They are infected with phage and transferred immediately to a medium containing light isotopes so that all constituents synthesized after infection are light. The distribution of new RNA and new protein, labeled with radioactive isotopes, is then followed by density gradient centrifugation of purified ribosomes. And so here we need to pause again to look at additional properties of ribosomes. Ribosomes take their names from the rate of sedimentation in Svedberg units, S, that is, how long it takes for the particle to pellet in a centrifuge at a given centrifugal force. And bacterial ribosomes have a sedimentation rate of 70S and are composed of 50S and 30S subunits, and you've probably noticed that centrifugal forces don't care about math. And it was found that a certain concentration of magnesium was required to keep ribosomes in their 70S complex. In low concentrations, ribosomes separate into their 30S and 50S subunits, but at higher concentrations, two 70S ribosomes aggregate into a 100S ribosome. And so Brenner, Jacob, and Meselson first looked at purified ribosomes and determined that they had a distinct biphasic shape, which they called the A band, which they found was a mix of 35S and 50S, and the B band, which was the 70S complex, and that newly synthesized RNA aligned only with the B band. And similar to what Nomura and Hall had found, this RNA band disappeared quickly after moving the bacteria to a non-radioactive broth, which indicated that it was unstable. They also determined that no new ribosomes were synthesized from either the host or the phage, and so these data effectively eliminated Model 1 since it required synthesis of new ribosomes. To address the other two models, the authors needed to determine if viral protein synthesis occurred on bacterial ribosomes or directly on viral DNA. So they grew bacteria in N15 media, which labeled all synthesized proteins, including ribosomes, and incubated them long enough for all proteins to be radioactively labeled. Then, they infected them with bacteriophage, moved them to a broth containing both N14, which is non-radioactive nitrogen, and S35 sulfate, which has radioactive sulfur, for 2 minutes, where newly synthesized proteins would have N14 and S35, and finally transferred the bacteria to media containing excess non-labeled sulfate and methionine to wash out the radioactive sulfate. They then purified the ribosomes and assessed if newly synthesized S35 labeled viral proteins were associated with pre-existing N15 labeled ribosomes, or if they were found in newly synthesized N14 viral ribosomes. And here we see the results. The arrows are where they expected to find non-radioactive newly synthesized ribosomes, and so we see that the peak of newly synthesized proteins, which are the black dots, coincide only with the B band of the pre-existing N15 labeled ribosomes, indicating that viral proteins are synthesized on bacterial ribosomes. And so the authors concluded that most, if not all, protein synthesis in the infected cell occurs in ribosomes. The experiment also shows that pre-existing ribosomes are used for synthesis and that no new ribosomes containing stable sulfur-35 are synthesized. This result effectively eliminates Model 2. It is a prediction of the hypothesis that the messenger RNA should be a simple copy of the gene, and its nucleotide composition should therefore correspond to that of DNA. One last point deserves emphasis. Although the details of the mechanism of information transfer by messenger are not clear, the experiments with phage-infected cells show unequivocally that information for protein synthesis cannot be encoded in the chemical sequence of the ribosomal RNA. Ribosomes are non-specialized structures which synthesize at a given time the protein dictated by the messenger they happen to contain. These results were extended to uninfected cells by James Watson and his group, whose findings were published back-to-back with Brenner's and provided evidence that this mechanism was a universal process. The same year, Hall and Spiegelman conclusively showed that mRNA was derived from DNA using a recently developed hybridization technique whereby double-stranded RNA exposed to heat separated into single strands and slowly cooling them reassembled them into their native double-stranded form. Hall and Spiegelman explain, such reconstitution of the double-stranded structure occurs only between DNA strands which originate from the same or closely related organisms. Presumably, the specificity requirement for a successful union of two strands reflects the need for a perfect or near-perfect complementarity of their nucleotide sequences. We have here then a possible method for detecting complementary nucleotide sequences. The formation of a double-stranded hybrid during a slow cooling of a mixture of two types of polynucleotide strands can be accepted as evidence for complementarity of the input strands. To determine if this might also apply to DNA-RNA double-stranded hybrids, Hall and Spiegelman radio-labeled single-stranded RNA with p32 and single-stranded DNA with H3 and mixed them with unlabeled double-stranded DNA and then centrifuged them in a cesium chloride gradient for 5 days to separate the molecules. And here we see that each species clearly separated with little overlap and provides therefore a test for interactions leading to the formation of RNA-DNA hybrids. Any distortion of the distribution of H3 DNA or p32 RNA from that observed in Figure 2, which leads to regions of overlap between H3 and p32, would be indicative of such interactions. So they mixed RNA with single-stranded DNA and allowed the mixture to slowly cool down over about 30 hours, and they found that slow cooling of the DNA and RNA has produced a new peak of p32 approximately centered on the band of H3, which is denatured DNA. This new p32 containing band must contain an RNA-DNA hybrid having approximately the same density as denatured DNA. They also determined that this peak didn't show up if the DNA and RNA were of different sources, confirming that the DNA and RNA sequences must be significantly identical for hybridization to occur. With these experiments, the authors concluded that the fact that RNA and DNA do satisfy the specificity requirement for hybridization must reflect a correspondence in structure between the two. Structural specificity of this order in single polynucleotide strands can only reside in definite sequences of nucleotides. We conclude that the most likely interrelationship of the nucleotide sequences of DNA and RNA is one which is complementary in terms of the scheme of hydrogen bonding proposed by Watson and Crick. The demonstration of sequence complementarity between homologous DNA and RNA is happily consistent with an attractively simple mechanism of informational RNA synthesis in which a single strand of DNA acts as a template for the polymerization of a complementary RNA strand. The same year, the authors showed that RNA in cells can hybridize to its own DNA, which is consistent with the assumption that DNA serves as a template for the synthesis of complementary informational RNA. This view would require the existence of a DNA-dependent enzymatic mechanism for synthesizing polyribonucleotide, and an enzyme with these characteristics, called RNA polymerase, had been discovered the previous year, and Spiegelman and his group concluded one of their papers this way. It would appear from the results summarized here that the synthesis of polyribonucleotide strands complementary to homologous DNA is a generalized feature of normal and virus-infected bacterial cells. With respect to the genetic transcription mechanism, the present data are consistent with the conclusions drawn from our previous experiments with T2-infected cells. It would appear that complementary RNA strands are the intermediaries between DNA and the protein synthesizing apparatus. In 1957, Francis Crick, who, if you remember from my video on DNA, elucidated the structure of DNA with James Watson, gave a talk on the biological replication of macromolecules in which he discussed the coding problem, that is, the problem of how, in outline, the sequence of 4 bases codes the sequence of the 20 amino acids. It had previously been suggested that 3 bases coded for each amino acid, that adjacent triplets of bases overlapped, and that more than one triplet of bases stood for a particular amino acid, which is called degeneracy. And though the first and third of these were likely by this point, though not conclusively shown, support for an overlapping code was tenuous. Assuming a triplet code, there are 3 possibilities. An overlapping code, where triplets coding for an amino acid advance 1 nucleotide at a time, a partially overlapping code, where triplets coding for amino acids advance 2 nucleotides at a time, or a non-overlapping code, where triplets coding for amino acids advance 3 nucleotides at a time, and nucleotides within a triplet are only used once. The elucidation of the coding problem was enabled by the discovery in 1960 of an enzyme that catalyzes the synthesis of polynucleotides, called polynucleotide phosphorylase, which allowed Nirenberg in his lab to synthesize specific RNA sequences to test the requirements of protein synthesis in bacteria. In a first experiment, they incubated polynucleotides composed of a single type of nucleotide, either uracil, adenine, cysteine, or inosinic acid, in a mixture capable of protein synthesis, along with radioactive phenylalanine, and found that only polyuracil could produce a polypeptide composed of a string of phenylalanines. They also conducted the reverse experiment, in which they added polyuracil to a mixture along with various amino acids, and found that only in the presence of phenylalanine could a polypeptide be produced. Later the same year, Ochoa's group found that phenylalanine could be incorporated into polypeptides using nucleic acids composed of combinations of uracil and cytosine, uracil and adenine, or uracil and guanine, as long as uracil was present in the higher ratio. In addition, they found that uracil and cytosine could also incorporate leucine, proline, and serine into polypeptides, whereas uracil and adenine also led to the incorporation of isoleucine, leucine, lysine, and tyrosine. These and other experiments allowed Ochoa and his group to first deduce 2 of the nucleotides required for incorporation of each amino acid, and then in subsequent papers, the combination of nucleotides required for incorporation of each of the 20 amino acids. However, the precise order of the nucleotides was still unknown, and the fact that the code was composed of triplet nucleotides was still merely an assumption. In 1964, Nirenberg wondered how long an mRNA would have to be for tRNA to associate with ribosomes, and his lab determined that if they passed a mixture of ribosomes, tRNA, mRNA, and a complex of all three components through filter paper, only the assembled complex was retained on the filter paper, while the individual components simply washed through. This gave them a much easier and faster way to isolate ribonucleotides, since we could do as much in one day as had previously taken us 8 to 10 days. Using this method, his lab determined that mRNAs required at least 3 nucleotides for binding of tRNAs to ribosomes, and that only multiples of 3 nucleotides increased the incorporation of amino acids into a growing polypeptide chain, providing direct proof that the genetic code was composed of non-overlapping triplet nucleotides. And so obviously, the next step was to synthesize all 64 possible combinations of triplets and determine which triplet codes for which amino acid, and this took about a year to accomplish, the genetic code being completely deciphered by 1966. In later studies, researchers found that the AUG codon, which codes for methionine, initiates the protein synthesis process, meaning that all proteins begin with this amino acid, whereas the codons UAA, UAG, and UGA do not code for any amino acids, but rather signals the end of the protein, at which point the ribosome dissociates and is released from the mRNA, along with the completed protein. Additional studies comparing the genetic codes of bacteria, invertebrates, and mammals found that this code was universal, and therefore the process of protein synthesis was essentially the same in all organisms. As Nirenberg stated in a review of the discovery of the genetic code, these results had a profound philosophical impact on me because they indicate that all forms of life on this planet use essentially the same language. Although practically nothing was known about DNA, the complex mechanism of protein synthesis, or the genetic code at the beginning of the 1950s, these were all fully elucidated less than 20 years later and confirmed that DNA was the site of storage of genetic information. However, that is the extent of its function. To be of any use, this molecular code needs to be transcribed to messenger RNA, a ribonucleotide with a base sequence complementary to the section of DNA from which it is derived, and which in turn is used as a template for protein synthesis on ribosomes. The initiation of protein synthesis was determined to begin after the small ribosome subunit, which is the 30th subunit in bacteria, binds to a recognition sequence near the beginning of the mRNA, and where it scans the mRNA, that is, it glides along the mRNA, reading its nucleotide sequence until it encounters a start codon, AUG, at which point the large ribosomal subunit, the 50S subunit in bacteria, binds to the complex, and this stimulates the incorporation of methionine as the first amino acid of all proteins. But before this can happen, methionine needs to be activated by ATP and incorporated into transfer RNA, which acts as an adapter molecule between amino acids and mRNA. A sequence of 3 nucleotides on the tRNA complex, which is dictated by the genetic code, forms hydrogen bonds with a complementary sequence on the mRNA. For example, methionine is coded by the triplet AUG, so the triplet on the tRNA would be UAC, since uracil pairs with adenine and cytosine pairs with guanine. After the activated amino acid binds to the mRNA on the ribosome, the amino acid is transferred from the tRNA to the growing peptide chain via a peptide bond. Amino acids continue to be added as dictated by the mRNA sequence, and synthesis ends when the ribosome encounters one of the 3 termination codons, at which point the ribosome complex disassembles and releases the mRNA and the protein. And so this brings this video to an end. I hope you enjoyed it, and if you did enjoy it and would like to know more about this topic, please check out my book, Landmark Experiments in Protein Science, using the link in the description. And if you have any questions, please leave them in the comments and I'll do my best to answer them. And I hope you'll join me for the next video, which will be on the mechanism of cell signaling.